Professor Baroud studied physics and mechanical engineering at MIT and then at University of Texas at Austin. Later did his postdoc at UNS. Then he has been holding a faculty position at École Polytechnique. He has worked on various aspects of microfluidics. Initially, he was focusing on the physical aspects, and recently he has moved on to biological applications. He is well known across the globe for his research on droplet microfluidics. He's also leading a research unit focusing on the use of droplet techniques to learn new biology. With that, I would once again invite Professor Charles Baroud and his postdoc, Svens, for the talk.
Thank you very much. Just there's somebody who has a microphone on. Maybe there's a lot of noise. That's much better. Can you hear me? All right, super.
I can hear you. I'm just trying to look for. I'm trying to mute several speakers.
Okay, super.
Now it seems fine.
Now I think it's
it's okay. So let me just share my screen, and I think this is what I have to do, if I remember correctly. Can you see my presentation?
Yeah, we can see it.
Super. So thanks again for the invitation. I'm actually very happy to be presenting in front of you today. And as I don't know if this is the title that I sent initially, but I will be talking to you about some of the work that we've done in my lab about doing 3D cultures and microfluidics based on droplet microfluidic techniques that we have developed in my group over the last 12 or so years. And so I thought that since I'm going to be presenting some of the new work that has been done by Shreyansh Jain, I asked him to also present this part rather than me presenting it for him. And so I look forward to this as well. Before going into the science, let me just tell you a few words about our group. So we are a group jointly between École Polytechnique and Institut Pasteur. So École Polytechnique is an engineering university in France. It's one of the elite schools here that attracts brilliant and strongly mathematical students. And Institut Pasteur is a biology institute. And so by being co-affiliated with the two, the objective in our work is to try to bring a quantitative and mathematical approach to biological problems. And we do this based mostly on microfluidics, although sometimes we also try to do other things. The group is about 12 people, more or less, coming from different backgrounds, from biology, physics, engineering, math, chemistry, and different things. So what type of problems do we work on? Well, from the biological applications point of view, we have basically two major areas that we look at. We look at microbiology problems, for example, looking at the development of colonies of bacteria that originate from an individual cell. So from a single cell, how do we make a colony of cells? And this under antibiotic stress or not. And so we're looking at the response to antibiotics from in these situations. In the microbiology field as well, we're looking at how, so this is the movie that you're seeing in the bottom, how does a single yeast cell repair its DNA and divide as it's repairing the DNA. So again, in all of these cases, we're looking at how, when you start from one single cell, how do you create a progeny that is not only dividing, but also responding to some kind of stress. And then maybe 2/3 of the lab are working on 3-dimensional cell culture. And so this is in wide variety of different subjects. So one of them is how do we actually make simply a spheroid or an organoid out of individual cells. So this is, I'm going to show you a lot about this today. I'm also going to show you, and Shreyansh is going to explain, how do T cells attack and kill cancer spheroids. So this is going to be the last part of the presentation. But in addition to these two subjects, Here, I won't show you the rest of this movie. So in addition to these two subjects, we're also working on the development of mouse embryo-like structures. So how do you start from a few embryonic stem cells and then allow them to develop and make something that looks almost like a mouse embryo? So in term of developmental biology. And then finally, another subject that was initiated by Shiansh also in the lab has been to take three-dimensional cultures like spheroids or organoids and to actually apply mechanical stress on them. And the idea here is to understand how does the three-dimensional structure of these biological objects influence how the mechanical forces are transmitted, and then in turn, how do these mechanical forces influence the cell phase? So this is a subject that's emerging in our lab that probably we will be working on in the next few years. So there's a wide variety of biological questions, but these biological questions have in common, first of all, an experimental and theoretical approach, which is through microfluidics and quantitative measurements. But there's also, beyond this kind of technical unity to the subject, there's also a question, kind of a philosophical question. So if you look at what is done typically in a lot of biology labs, then what you find is that there are two scales that are kind of in opposition with each other. One of the scales is the population scale. This is what you do when you put just a bunch of bacteria in a tube and you let them grow. And the other scale is to look individually at single cells and to really try to dissect individually how the cells are behaving. So at the population scale, it's relatively easy scale to work on. You get an answer directly because, if you want to know if the antibiotic is killing the cells or not, well, you just put the antibiotic and you look and you can see if it's working or not. Relatively easy to analyze and manipulate, and they're relatively insensitive to heterogeneities in the populations. And this is because in a large population, the answer that you get is dominated by it's dominated by the dominant subpopulation. So if you have a subpopulation that is less fit or less or behaving a little bit differently, you typically will not see its influence. At the single cell scale, the idea here is usually to gain a deeper understanding of the mechanisms that underlie the global behavior. You want to see the heterogeneity on the cellular level. And what's important is that each one of the cells will behave not in the same way as its neighbors, but it will depend on its local environment. So actually, what the answer that you will get for each one of the cells depends not only on the cell itself, but it depends on the very local environment around the cell through mechanical or chemical or biological cues. But there's a tension between these two scales. The strong advantage is to bulk methods. If you look at any biology lab in the world, what you will see are PCR machines and culture platforms and so on that work on bulk scale, that work on population scale. And maybe most of the single cell measurements is done through single cell imaging and flow cytometry, even though this is starting to change now with some of the modern sequencing techniques. But linking these two scales is difficult. It's difficult for many reasons. One of the reasons is that the population is not just a collection of individual cells. You need to account for interactions between these cells. And so now you need to take into account not only the individual cells, but their interactions. And this becomes a very large space to explore. And time and space both play an important role. So things evolve in time, also in space, and you have to account for both of these dimensions. So there's an intermediate scale that is really the scale that interests us in the lab. And I don't know how to call this scale. And the best name I came up with was the collective scale. At the collective scale, we're going to look at starting from 2 cells that can interact with each other to maybe 10,000 cells. At these scales, you conserve the sensitivity to the heterogeneity between the cells, but you can start to look at the variability and also the interactions between the cells. And so this is a scale that's largely unexplored. There are two difficulties, I think, to exploring this. First of all, there's a technological gap. There are very few tools that are actually able to address questions at this collective scale. And this is part of what we have been doing in microfluidics. And then once you measure things at this scale, because these are new measurements, there are very few conceptual tools. So there are very few models and mathematical descriptions of how things behave at this scale. And again, this is kind of a snapshot. Things are evolving. There are new things every day. But if we were talking 10 years ago, then the answer would have been very, very few. And now there's still very few things that are available. However, of course, people have been working at this scale in different subjects. So in microbiology, for example, a lot of the work on biofilms is trying to bridge the gap between individual cells and biofilms. And you can find actually some really nice work in systems biology, for example, on trying to understand interactions within the bacterial population that lead, that make the connection between what the individual cells are doing and how things happen in biofilms. So this is one example that where things are developing. Another example in mammalian cells is how do you bridge, single cell measurements with organisms, and a very exciting field that's being developed right now is the field of organoids and organ-on-a-chip, where you start to build structures that account for the spatial structure, the variety of different cells that are interacting together, as well as spatial temporal behaviors and maybe mechanical forces and chemical gradients and so on. So this is really a very, very hot topic where the questions that are being asked today are simply questions that were not possible to ask maybe 10 years ago or so. So this is the general context. This is the kind of the big picture of what we're trying to do. So by the way, if there are any questions, I'd be really happy if you interrupt me for questions. Otherwise, I'm just speaking to a screen and I don't know if anybody's listening. Right, so this is where we're working. We're working at this collective scale. How are we doing it? Well, mostly through microfluidics, as I said. And so my view of microfluidics is that what we want to do is we want to reduce the volumes that we're working on, and we want to integrate many operations on a single device. So the integration part is something that already exists with these multi-well plates that are well developed. And then the miniaturization part is also something that already exists. And so this is a picture of a microfluidic device from Fluidigm. This is something you can buy. It exists, it works. And it's quite old now. It's more than 20 years old. But the important thing in this view of microfluidics is that by by integrating a large number of replicates, not only can you just get more data, but you can ask new types of questions. This is what we're going to do. And what we're going to work on in particular is we want to replace this microfabricated chambers with valves and pumps by droplets. And what I mean by this is that we're going to use water drops in oil or aqueous drops in oil. And then if we control properly the chemistry of the oil and the water and so on, then we can consider each one of the droplets as being independent of its neighbors. And in this way, we can run a separate reaction inside each one of the droplets. And this has the advantage of being much easier to do technically than the complicated Fluidigm chips. It can be scaled. We can do a lot of these droplets, and we can follow the contents of the droplets in time. And this is what we've been working on for a long time. Okay, and so maybe the last part of the introduction is that what kind of biology are we going to address, at least in today's presentation? We're going to address three-dimensional cell culture. And so three-dimensional cell culture is interesting because as you know, in our body, the cells are in contact with each other, and they're almost never in contact with a plastic, hard plastic dish, at least not for any long periods of time. And so what you would like to reproduce in the biological models that you're doing is cells that are only in contact with other cells. And so for this, kind of the classic way to do this is to make spheroids. And so spheroids are collections of cells that are only in contact with other cells. And the classic way to make spheroids is to put the cells inside of a liquid droplet, to let them sediment by gravity, and then they aggregate together. But then that's not still a spheroid, that's an aggregate, but the cells will make junctions between each other, and they will organize themselves inside the three-dimensional structure. And so the spheroid is what happens after the cells have been allowed to actually really make intimate junctions with each other and to organize. And this again is a technology that exists now. So it exists from the US or in Switzerland. You can buy plates to make spheroids, to observe them, to add drugs, and so on and so forth. So this is all kind of the background. Now, what are we actually going to do? One of the questions we are going to ask is, can we make a multi-well plate based on droplet microfluidics? Can we perform 3D cell culture inside it? And then from this, can we get really detailed cell data, single cell data, in order to learn new biology? So this is kind of the long-term picture that we've been developing. I'm going to show you this by first introducing the two-dimensional droplet microfluidics technology that we've developed, the applications to cell culture, and then showing you a little bit the new biology. Okay, so this is the physics part of the talk. When you make a droplet, so let's say a liquid, a water droplet in oil, for example, the volume of the droplet is fixed, unless you lose some volume, but typically you consider the volume as being fixed, but the surface area around the droplet is not fixed. Actually, the surface area depends on the shape of the drop. It is minimum for a sphere. This is why droplets want to make spheres, because they want to minimize the surface area which costs energy. So the surface energy, which is proportional to the surface area, changes depending on the shape of the droplet. So now if we take a drop and we squeeze it between two walls, we will increase its surface area. And this is what you see in the green drop is supposed to represent, for example, water, and the orange is supposed to represent oil. And this is a cross-section of a channel. So in cross-section, when we squeeze the droplet, we make a pancake shape. And this pancake shape has a higher energy than if it was spherical. So if we now allow little places in the microfluidic device where the droplets can actually enter into a groove or something like this, then by doing so, they will reduce their surface area, and therefore they will reduce the surface energy that they store. And so because there's a lower surface, I can't move my mouse, I don't know if you see. Because the surface energy is lower when the droplet enters into the hole than outside, there's a force that's actually attracting the droplet to the minimum of surface energy. So this is a picture just in a cross-section, but actually we make these devices using microfabrication techniques. And so since we're using a photomask to make these, we can make any shape we want, really. And so in this way, if we make a droplet that is squeezed by the top and bottom walls, but that's sitting on a groove, then it will be attracted by this groove because of the surface energy reduction. And if we're pushing it at the same time with the external fluid, then it will move in the direction that it's being pushed in, but it will also follow the groove that we fabricated. This was the idea that we had a few years ago, and we can verify this. What you're seeing here is a water drop in oil. It's squeezed by the top and bottom walls, and it actually, there's a groove on the top wall, which is, as you see, much smaller than the size of the droplet. But even though the tuple is, even though the groove is much smaller, and we're pushing this droplet from left to the right with the oil, and what you see is that it indeed moves from the left to the right because it's being pushed, but it's also being guided laterally by the structure that we call a rail. And so this is interesting because now we're removing the sidewalls of the microchannel, but we're still able to manipulate the droplets laterally without using the sidewalls. And it turns out that this is a really useful operation because it makes these devices much more robust in practice. So as you can see, actually at the bottom of this picture, there's some dust in the channel, but even though there is dust here, the channel behaves perfectly well. And so the ability to remove the sidewalls actually makes the microfluidic devices much more robust to use. Okay, so this is a rail. Now what happens if we make just a single hole in the channel? Well, we get what we call an anchor. And so here is one anchor where we're attaching one drop. The drop is attached because of the surface energy gradient, and the other drops are just flowing past it. So this is one anchor, but as I said, we are making this using microfabrication techniques. So if we can make one anchor, we can make many of them. And so this is an example of many, many anchors in a similarly shaped device. And when we bring many droplets above them, then the drops just stick in place. And as you can see, it's a random distribution of droplets, but very easily we can make an array of droplets that are stationary. And this is something that we can now do every day in the lab. Is it clear? Is there anybody there?
Yes, I think they would ask questions and by typing it in the message box. But yes, yeah.
Okay, good. Well, I'm happy to see somebody's listening.
Yeah,
Right.
indeed. Yeah.
So the technology itself is very, very simple in the end. I mean, you have to manage the chemistry with the surfactants, the oils, and so on. But once you have this, then it's very easy to actually get this to work. Now the question that we ask is, can we actually perform cell culture inside of these droplets? The way that we were going to do this, so the objective we placed a few years ago was to make an integrated platform in which we can actually do a lot of operations on cells. So compared to many different droplet microfluidic techniques, the idea here is to work with stationary droplets that are not moving most of the time, and to do all of the operations inside of these stationary droplets, because it just seems easier to me to do that. So what we want to do is to produce the drops, to culture, to make the spheroids inside them, culture them and maintain them, change their environment, stain them and observe them, and potentially recover them, and do all of this in a single piece of plastic that's easy to make, easy to fabricate. So this is how it's going to work. Instead of most of the devices I will show you are based on this similar device, on a similar design, where we have one region that's being used to make droplets, similarly to what other people have done. We have a second region that allows us to distribute the droplets just to make the devices more efficient. And then a third region, which is used to trap the drops and observe them as a function of time. So rather than going into detail about describing the device, let me show you a movie of how this works. What you will see in the movie is starting from the third position from the right. And on the outside here is the oil phase. On the inside is the water phase. And you can see the interface between the two. And what you see are cells that are suspended in the aqueous phase. So when I start the movie, this will be in real time. So 1 second of the movie time is 1 second of the experiment. and okay, so here we go. 1, 2, 3, go. What you see is we start pushing the aqueous phase and we generate the droplets very efficiently at this junction. And we've spent a lot of time to make sure that the drops are homogeneous in size and have the right number of cells inside them. But in the end, what happens is you make a train of droplets that are being distributed now with these sideway rails. And the droplets arrive in the microfluidic chamber, in the culture chamber, where we have anchors. And so what you see is that in a few seconds, so in 30 seconds, we start placing the individual droplets inside individual anchors. The cells are free to kind of float around the droplets because of the flow. So what's important is that each one of the anchors is made so that we have exactly one droplet inside it. Sorry, I have a bit of a cold.
Okay, it's okay.
Okay, so each anchor has exactly one droplet inside it. But each droplet can contain about 100 cells or 150 cells. So by working with the cell density, we are sure that each one of the droplets contains some cells inside it. And we have exactly one droplet in each anchor. What you also see is that it takes less than a minute to start filling the chip. And so in total, maybe it takes 5 or 10 minutes to fill the whole chip. And once we filled the chip, then we can just remove the tubes and put it in the incubator. And then we just allow the cells to behave biologically. So now the next behavior is not due to the flow, but it's due to the biology. And so in this movie, what you will see is a time-lapse movie over, I think, 24 hours or 12 hours, but we'll see, where you have the cells.
Sorry to interrupt, Charles. There is a question in the chat box. Maybe if Ashish, if he would like to read it to you.
Yeah.
Perfect.
So
Sorry.
the question is, I think, regarding the movie that you showed in the previous slide.
Yeah.
Question is there a surfactant-based stabilization happening there?
Yeah, absolutely. This is very important. You really need to have the surfactants, otherwise the droplets, well, you have two problems if you don't have surfactants. The first problem is that the droplets can merge together. So maybe I'll show the movie again. What you see, when you make the droplets, they can touch each other. Well, not here, but once they exit this part, you see they're touching each other. So if you don't have surfactants, then the droplets can merge together, and then you lose a lot of the control that you have. And the second problem is that if you don't have surfactants, you can have wetting issues between the aqueous, the oil, and the PDMS. And you want to avoid this as much as possible. So you have to have surfactants in these systems, absolutely. Okay, so now once the cells are inside of the anchors, then we're going to let them sediment to the bottom. And then as they sediment, they will meet each other. And they will start to actually create junctions between each other. And as they create the junctions, you see that they start making a much more compact sphere of cells. And indeed, there's a moment at which they go from being really an aggregate of kind of loosely packed cells around now into something that's very, very smooth on the outside. And you feel like all of the cells have really connected with each other. And so, so we make the droplets and then we incubate the chip without any flow. And during this period, the cells will compact and make a compact sphere. So the time required to make these spheres depends very much on the type of cells that we're using. It can go from about 5 hours to about 24 hours or more for certain cell types. And then there's a really cool trick that we can play, which is that since these are spheres in aqueous solution, we can make a flow. And by flowing the fluid, we can basically tumble the spheres and make sure that really they are really spheres. We can see that the cells are nicely adherent together, and it's just a nice way to check that everything is what we think it is. Okay, so this is how we make the 3D cultures, the spheroids. Now we have a few problems. One of the problems is that there's a limited amount of nutrients inside of the droplets because of the limited volume. And also we cannot really bring anything more to the droplets because they're surrounded by oil. So one way to solve this problem is to make the drops with a gel inside them. So this gel can be either an inert gel like agarose or a biologically active gel like collagen or matrigel. If we do this, we make the spheroids and then we gel the droplets. And then what happens is we can remove the oil and replace it with an aqueous medium. And this is what it looks like with an agarose gel. You see the spheroids are in the agarose, and we are able to remove the oil and replace it with an aqueous medium. And the spheroids stay nicely in place. What can we use this for? Well, we can use this first of all to bring culture medium if we want to add nutrients or to wash, byproducts. We can use this to bring drugs or any kind of antibody or liposomes to interact with the cells. We can also use it to bring antibodies to do the immunocytochemistry, basically to mark different proteins in the cells so that we can do the microscopy on them. And indeed, we can bring any water-soluble molecule and it will just diffuse typically through the gel. And so what we get in the end is that we get images like this, where we have the spheroids and we make several staining and washing steps inside of the microfluidic device. And so we can permeabilize the cells, fix the proteins on them, bring the first antibody wash, second antibody wash, and so on and so forth. And what we get in the end is on a single device, we can get a single cell imaging on each one of the spheroids. Depending on the device, we have a few hundred to maybe 100 to 500 spheroids on a single device. And because of the imaging, we get single cell resolution on this. And then what's necessary is an image analysis pipeline. But if you have this image analysis pipeline, you can really build what we call multiscale cytometry. So cytometry means that we're measuring things on cells, so on individual cells in particular, and multiscale because we are able to measure things on the population level, on the spheroid level, and on the single cell level. And most importantly, we're able to relate what we measure on each individual cell with its position in the spheroid or around it, and the spheroid with its position within the population. And so this, I think, is a very, very powerful idea that allows us to bridge what I was telling you initially, the single cell measurements with the population measurements. I think there's a question in the chat. Let me see if I can open it.
Yeah.
Cells don't need to adhere to a surface to grow into a spheroid. Yeah, so this is the whole point. The whole point is that these are cells that adhere to each other. And because they are adhering to each other, they don't need to adhere to a plastic surface. And as a matter of fact, the adhesion between the cells is much more physiologically relevant than adhesion to a hard substrate. So not all cells are able to make spheroids. Sometimes we need to add some extracellular matrix to the mix. Sometimes they really don't make spheroids, but a lot of different cell types are able to make spheroids. Sometimes they grow, sometimes they don't, but that's biology. So there is no general answer, but a lot of different cell types are able to make spheroids. Right. So what do you do with this? Well, what do you do with this is that then you develop a very detailed image analysis platform. So this is something that's still being developed in the lab, where on each one of the spheroids, you want to identify the positions of the cells. You want to identify what the cells are producing biologically and how are they connected to each other. So this is really ongoing work in the lab, but we're working towards many ways of doing this to answer specific biological questions. And so what you get from this is that you can do population level measurements, like for example, ELISA, or you can recover the spheroids to do a PCR on them and any standard biological technique. You can also ask questions on the scale of individual spheroids, and you can get cellular level measurements. So now, okay, I should speed up a little bit so that I leave Shreyansh with enough time to present his work. But so the other problem that we have so far is that the same thing is happening on all of the cells. What we would like to do is have different operations happening at different positions. And we do this by making anchors that are a little bit differently shaped. So we add this little nose on the edge of the anchors. And by doing so, even when we have an initial droplet in the major part of the anchor, we leave enough space to bring a second droplet later on. This really works. So we can make a first droplet that fills the big part of the anchor, and then at a later time bring a second drop. And then by removing the surfactants, we can actually merge these droplets together. And so here what you see are droplets that contain food dye, so nothing very fancy, but just food coloring. And then we can make a first population of droplets that go from green to blue to yellow, and then a second one that go from colorless to deep red. And when we merge them together, we create all of the different combinations of these different colors. And in this way, we can explore the red, green, and blue space just simply by merging droplets together. So in practice, what we want to do is we want to make spheroids in the large droplets and then use the small droplets to bring a second species. So there's another question. What's the mechanism by which the droplets get attached to the anchors? Well, it's the same mechanism as I told you before, that the anchors are actually holes inside of in the ceiling or in the bottom of the microfluidic channel. And since the droplets are squeezed elsewhere, they actually go into the anchor. So I will show you a better picture of this, which hopefully will explain this better. So there's a picture in 2 slides that will show this. Right, so a few applications of this idea. The first application is to make a spheroid in the first droplet, and then to bring a second droplet that contains beads that are functionalized to attach certain molecules. And so then when we merge these things together and we use hydrogels, what we are able to do is to attach on these beads whatever is being secreted by the spheroid. And so these are spheroids of mesenchymal stem cells. They secrete many molecules, and one of these molecules is called VEGF. And so if we bring beads that are functionalized with anti-VEGF antibodies, after the fusion, what we're able to do is we're able to capture the VEGF on the beads, and then we also bring the second antibody that will bind to VEGF, and so the beads become more and more fluorescent the more VEGF is attached to them. And so in this way, by determining when we merge the beads with the spheroid droplets, we're able to measure how much each one of the spheroids has been able to secrete in terms of these secreted molecules. So we make measurements on individual spheroids, and this is already kind of a technological challenge that we're able to do. But what's going to be more interesting is to have more complex biological operations. So this is a cross-section of these anchors. And this is what I was saying is how the droplets get attached. So the droplets, when they are not in the anchor, they are squeezed by the top and bottom. And when they reach the anchor, they can become spherical. And so they stay in there. So what we do in these experiments is that we bring the first droplet, we make it, we anchor it in the first anchor, and we give it a day for the spheroids to form inside it. And then at the second day, we can bring a second droplet. We can make a spheroid in the second droplet. And then by merging the droplets together, the spheroids come together. And so in this way, this is what it looks like. This is a big droplet that has one spheroid and the second droplet that has a second one. And when we merge the droplets together, the two spheroids come together and they make a single spheroid with 2 cell types. And then we can repeat this operation again. And by repeating this operation again, we can make now a tissue with 3 cell types in it. And this is done in each one of the positions in which we have a spheroid. And so step by step, we can build different heterospheroids. Now here I'm cheating a little bit because these cells are all the same, they're just colored differently. But we can actually bring different cell types, and depending on the combination of cell types, then we're able to actually make systems that have different, a core shell structure or side-by-side structure or different types of structures. These were synergetic operations. Here we have antagonistic cells, so we can actually look at host pathogen interactions by bringing bacteria in contact with spheroids and see how the bacteria are able to kill the mammalian cells. Or we can bring immune cells that are going to attack cancer cells. And then finally, we can also bring not cells in the second droplet, but molecules. And so in this way, we're able to bring different concentration of drugs in each one of the positions. And by barcoding the droplets, depending on what's their contents, we can make a complete drug screen on a single device. So on a single device, we have 250 spheroids, and we have, so in principle, 250 different concentrations of drug. And we're able to follow this in time. And what we see is the following. So what we see is that as a function of the drug concentration, for low concentrations, we have very high viability of the spheroids. We can measure the viability using molecular markers. For high concentrations, we have very low variability. So we have the standard drug response curve. And then between the two, we have a transition, which is what we would expect, except that we're doing this on 3D culture, and we're doing it in a single microfluidic device. Okay, so I see that I touched the nerve here, so there's many questions. So let me try to address them.
Yeah, I think the question is how the drops are being merged because they are surfactant stabilized.
Yeah.
And how to remove the surfactants from the droplets.
So this is kind of the nice thing about working in a microfluidic device, is that we can actually change the surrounding oil very easily. We just connect it to a different syringe and we bring a different oil. So in practice, what we do, we have a few techniques, but the one that we use most often is that we replace the very strong surfactant that we used initially with a weaker surfactant. So the second molecule is called perfluorooctanol. And so by replacing the strong surfactant with a weak surfactant, it replaces it on the interface. And what happens is that it's a less good surfactant in stabilizing the drops, so the drops just merge together. But since the drops are individually in different anchors, we don't mix the contents of different drops, we just merge the drops that are touching each other. And so this is a way that reliably allows us to merge droplets together. And then we can, again, wash the surfactants by bringing pure oil and so on and so forth. So the protocols may be a little bit more complicated, but that's the basic idea.
There's one more question here. In fact, there's the question I also had. Is there, given the low volume of droplet, is the limited concentration of nutrients not a problem? Is there a ratio of, the anticipated spheroid size to drop size? That is...
Yeah, so this is a very important question that we're continuing to address right now. And there are several ways to answer this question. The first way to answer the question is that if you look at these graphs that I'm showing on the screen right now, what you see is that in the absence of drug, or for low concentrations of drug, the spheroids are perfectly viable. So at least for this cell type, this droplet size, and these conditions, the spheroids are viable up to 30 hours. And so in a sense, we have an internal control inside each one of the devices that tells us that for this system, we're not limited by the amount of nutrients. The second way to answer this question is that this is going to be very cell type dependent. So some cell types, in particular stem cells, so either iPS cells or embryonic stem cells, they have very high metabolism. And in that case, you have to worry about the volume. And so we are working with larger volumes, or we are changing the media more frequently. So this indeed is very important. And then really to answer this question more in detail, We have a paper that's already online in chemical reviews, where we address this question in a lot of detail. You know, how much time can you culture cells inside of the 3D droplets, and so on and so forth. So if you look at chemical reviews, Shreyansh is one of the authors of the paper, and so am I. We address this question in a lot of detail.
I see there are 2 more questions. Would you like to answer those as well? No?
Yeah, so
I think this question is about when you mix different cell types, do they mix the way they are added, or there is some active cell rearrangements?
There is active cell rearrangement. What we think is that the final state will be mostly determined by the cell types. So this is a kind of an obscure question in cell culture, that, you know, where do cells actually place themselves in 3 dimensions? There are many factors that play, but for sure the nature of the cells, how adhesive they are, how rigid they are, and so on, will be important in determining where they place themselves in the 3D culture. So they sort themselves based on their biophysical properties rather than the order in which we bring them. And then the last question, I guess, is about the surfactants. So most of the surfactants are in the oil phase. The cells typically don't like being in contact with surfactants, so we avoid putting surfactants in the aqueous phase. Good. So I'm happy that I'm happy to receive all of these questions. I hope I answered them. Right. So this is the drug screen. I'll skip through this because it's not super important. So now I'll switch over to Shreyansh, who's going to tell you about the kind of the exciting work that we've been doing recently on immunotherapy. So wait, there's just, is there any other method to release the trapped droplets other than high flow rates? Let me get back to this later. Let me get back to this at the end of the presentation. Okay, so Shreyansh, you want to present the immuno-oncology work?
Okay, sure. So I don't know if host can pin me somewhere so that it doesn't appear that there is a recorded voice coming for the presentation. Thank you, Charles, for including me for the part of the presentation, and thank you for setting the context about the droplet microfluidics and the potential uses of it. So what I would be telling you today is one of the potential use of these cell cultures in the droplets in the field of immuno-oncology. So as you know that in the field of immuno-oncology, there are immune cells which are trained to specifically go identify an antigen and kill the cancer cells in the tumor. Now, what we try to do using this assay is to discover or to identify the dynamics of this immune cell attacking and killing the cancer cells inside the tumor. So what we do here is we make, as Charles explained, a spheroid from melanoma cells. So these are from skin cancer. They are B16 cells as they are known. And what we also do is something to add matrigel inside these liquid droplets, because you want these immune cells eventually, which will be added later on, to go and migrate inside these droplets. So if you can go next, Charles. Yeah. So again, what Charles explained to you before is you can bring a secondary droplet. So what we do here is a day later, once the spheroids are formed, we bring the secondary droplet, which contains immune cells. Now it's important to mention here that these immune cells are already primed in the sense that they are already trained to go to recognize this particular cancer spheroid, which is inside. So it's kind of a model system where you can study the dynamics of immune cells, and you are sure that where they're in contact, they will probably have some immune synapse and potentially some reaction to start killing the cancer cells. So what you can do is further you can fuse these droplets, and once you fuse these droplets, you can obtain your tumor spheroid sitting in the center, and then your immune cells, which are just fused somewhere in the place in the corner of the droplet after the fusion. And it's important to note here that when we fuse these immune cells, we can get variety of the immune cell distribution in terms of their number inside the chip. So if we go next, so what we can do using this model assay is first thing is we can track the migration of these immune cells, and you can track in terms of their history and how spatiotemporally they have evolved. You can see some immune cells which comes and gets attached to the spheroids. I think, Charles, you have to play the movie. So yeah, you can see the immune cells are marked in green. It's a GFP label. So they can come, get attached on the spheroid in this process. So you can see this in a very good high resolution. If you go next, is you can, of course, do some quantification of their velocity dynamics and obtain some relevant numbers. So what you see here is a very important number, which we found that the immune cells, when they're migrating inside the matrigel, their speed is higher. However, when they get attached on the little spheroid, they kind of tend to get slower down. And of course, it could owe to the reason that they come, they attach, they start forming a junction. Specifically, it's called immune synapse in this case, which is important for immune cell to eventually destroy the cancer cells. So this is the simple thing which we can identify using this. Now, as I mentioned, that these B16 cells, these melanoma cells, have a specific antigen which can be recognized by these T cells. But what if there is no specific antigen present outside these melanoma cells? So you can see these two clear dynamics, where in B16 ova, where this antigen is ova will be, the T cells can come, recognize, get attached on your left, and then can destroy or start destroying the tumor cells. However, in B16 wild type case, where this antigen is not present on the tumor cells, there is no accumulation, no attachment, and potentially no killing happening in the time frame. So this is a specific interaction. And what we see in the case where T cells are attaching, recognizing the antigen, and eventually destroying the tumor, is they start their numbers on the tumor starts to go higher. So over the time frame, you can see that the T cell number increases on the tumor. However, in the wild type case, there is no such accumulation of the T cells. If you go next, now it's important for us to also see if this kind of tumor or cancer cell death is a programmed cell death. And you can obviously see it using some specific death markers like caspase-37 in this case. So it is ensuring that the cells are not dying, as somebody asked above, like potentially due to necrosis or some nutrient deficiency. So it's pretty much coming from a programmed cell death because of the cytotoxic effect of the T cells. And you can see this caspase-37 signal evolves as the tumor dies. There is a simple way to tell if the cell is dying or not, is by just observing in the bright field the fragmentation of these debris, which cancer cells kind of produces when they are dying. So we use this as a simple marker to tell if the cancer cells were dying or not dying. Now, once this basic groundwork is set, we are sure that there is a specific death happening. But we can now start asking that what are the steps towards a successful tumor regression. Now, as you can see that there could be potentially 3 phases of it. First, the T cells, or let's say here it's a cytotoxic T lymphocytes, CTLs, they have to find this spheroid, the tumor. Then second case would be they have to accumulate on this spheroid. And then the third phase, as you can recall from the movies which you have seen earlier, is that they have to destroy successfully the cancer cells in this spheroid. Now, with these few questions, we started to first look at the overall dynamics of many of such experiments, which can be run parallelly in this kind of device, which Charles explained you. We asked this if tumor regression or the cancer cell death is always happening in a particular time frame. So in this movie, you see that it's arranged in the increasing number of the T cells inside the droplet. And you can see that in the top row, there is a lesser number of T cells. However, in the last row, there is a more number of T cells. And you can see that in the same time frame, however, when most of the cancer cells in the last rows are dying, some cancer cells don't die eventually if there are fewer number of T cells. So we asked if we can find or we can recapitulate these things through a modeling perspective, and also doing so, if we can find out some basic biophysical parameters which are involved in this tumor regression process. So here we found a simple number which we can map by just recording how many tumors the cancer cell starts to die, let's say at a time frame of 14 hours, and how many didn't. And if we can map it with the number of CTLs present in the droplet. So what we found is some number like this, where all the cancer cells or all the tumors where we can see a first cancer cell death event happening, there is a higher number of T cells associated with it. So you can see that somewhere above a particular threshold number of T cells, all the tumors show some cancer cell death happening in that particular time frame. Below a particular threshold, you see that most of the tumors, they survive. However, there is a very interesting band, which is somewhere between 7 spheroid number on the tumor and 3 CTL numbers. You can see that sometimes a cancer cell would die in the tumor, but sometimes it won't. And this is something interesting for us to see if these kind of behavior can be mapped biophysically, and if it can give us more detail about the dynamics of these T cell mediated cancer cell killing. So if you go next. So as I explained before, in the process, you can see that some T cells will come, they will attach on the tumor. However, some T cells can leave the tumor site as well, as you can see in the movie bottom. So it's basically a two-step process where cells can come, attach, or it could leave, and you can map it over the entire time frame. As you can see that at a high spatial resolution, you can see the cells coming and attaching. So you can easily map it, how it evolves over the time. Using a Markovian chain method, you can further analyze these kind of behavior and identify an incoming rate or outgoing rate of a CTL. Let's call it lambda in and lambda out, which is the incoming rate of a CTL and outgoing rate of a CTL, given the condition that how many number of CTLs were already present on the spheroid. And you will see there is something interesting coming out of this kind of quantification. So first, it was something which we saw or we asked if how the first CTL or the first T cell finds the tumor or kind of reaches to the region of the tumor. And we found that it is very consistent with the randomly arrival of some particles diffusing inside a droplet. So it's pretty much a random process, how the first CTL comes. However, when we start looking at the arrival rate, lambda in, or let's say detachment rate of lambda out of the CTLs, given that there was already, let's say, 0 number of CTL present. So then you have the arrival rate of 1. Let's say if you have 1 CTL, which was already present on the tumor, then what is the arrival rate of the next one? You can see that it increases with some amount. If there were 2 CTLs already present on the tumor site, the probability or the arrival rate of the third CTL to come is further higher. And eventually with the 3 CTLs already present, it's even higher and higher. So this is a kind of accelerated arrival rate of the CTLs, given that there was 1 CTL present, 2 CTL present, or 3 CTL present on the spheroid. So this is a kind of signature of a positive feedback loop. And there is something which is kind of enhancing the CTL arrival rate with the time and with the number of CTLs present on this spheroid. When we were doing this work, we found that there was a study which was published, which hint or which kind of showed that there could be some chemokines which are involved in this process of rapid accumulation of the CTLs. And I would not go much detail into the biology of it, but there are some receptors on the cell which can identify some particular chemokines which are being released from a distance. So this is kind of a long range process where CTLs, which are already present on the tumor site, can call far off the CTLs and can initiate an accumulation process, a rapidly driven accumulation process. So once this long reach process is kind of happening, we asked if there is a local or a short range process happening at the tumor site locally. So we started looking at the tumor specific region and the CTLs present on the tumor. And we found that T cells always tend to accumulate in some particular site of the tumor, and then they produce some death of cancer cells at that region. We found that roughly we found 3 or more number of CTLs present in that region, local region, which is producing the death. So there is some local cooperativity of the CTLs on the tumor site specifically. So we asked if we can model this to understand more biophysically into this process. So what we did is we modeled the spheroid fragmentation. So as you saw that some spheroids, some cancer cells can fragment out during the death. So we kind of modeled this spheroid fragmentation rate by considering that all the T cells are independent in their activity. So it's like one cell comes, kills, but then it is independent to the killing efficiency of the other T cell in the vicinity. So when we modeled it using a simple formalism, we saw that there is a parabolic profile of these fragmentation rates with the increasing number of T cells. So as the T cell number increases, you see there is not a rapid increase in the fragmentation probability. But however, experimentally, what we see is a very nice exponential increase in the fragmentation rates with the T cell number. That means it's not the independent activity of the T cells which can determine the fragmentation probability of the cancer cells, but it is something more to the independent activity. And we reasoned it that there must be this local cooperativity effect which enhance or drives the increase in fragmentation probabilities or fragmentation rates which we have observed experimentally. Now combined together, this effect of long range and short range, what we tried to map or what we tried to propose is a unified picture of how we can identify these fragmentations of cancer cells on the tumor. So as you know that we can obviously derive this incoming and outgoing rate of the CTLs on this spheroid, you can use these rates and can simulate over the time the number of CTLs on this spheroid. Once you know the number of CTLs on this spheroid, you can definitely identify the fragmentation rate. As I mentioned before that we have formulated a cooperative effect to see how we can identify the fragmentation rates on the tumor. So combining these two, we can have a complete picture of how the spheroid fragmentation probability evolves with the number of CTLs in the droplet. And if you can recall some slides before, I mentioned that there is a particular band in which there is a probability of the tumor or the cancer cells to either die or to survive. And using these two long range and short range biophysical parameters, we could successfully recapitulate this dynamics of the tumor cell or cancer cell fragmentation. So overall, as you can see, even though I brushed up quickly on this work, overall you can see the power of having the large statistics using this simple small device where you can run hundreds of such parallel experiments, though independent experiments, and how these kind of large statistics can help us bring a more realistic model where we can identify some kind of biophysical effects, which can set us a ground to more look deep into the biomolecular effects of what could be happening. So now I would like Charles to kind of summarize over the entire presentation. Thank you.
Friends, I have a question.
Sure.
T cells, do they start destroying from the surface of the spheroid and go inside, or they can really invade and do it randomly as they wish.
Okay, so we have qualitative insights to this. So what we see that T cells go, they attach on the surface, they start killing on the surface. But then you can see that in some movies, the tumor is completely destroyed. So maybe it's like a killing which starts from the surface, and then eventually the cells keep on going inside as they eventually kill from the surface towards the center. But probably if there is a penetration of T cells inside the tumor thing, I mean, I think maybe or maybe not.
Okay, so there's one more question in the window, if you can.
Yeah, sure. I see. Will the cooperative effect be enhanced with smaller droplets because of increase in local concentration of chemokine? So yes, this is a very interesting question. So you can think about this in two ways. So it can increase, or let's say it can prepone the activity when it begins the killing thing, because you know, in the small droplet, the gradients will be established sooner. But then again, what could happen is these gradients of chemokines could saturate quickly in a small droplet. So we don't know how this could interplay into the killing dynamics, but definitely the droplet size could play some role in terms of how quickly it could start happening, the cancer cell death in the time frame.
So also this is something that we're working on actively right now. There's a new PhD student in the lab who's trying different droplet sizes to see if we recover the same dynamics. Okay, so we've been a little bit long, but hopefully interesting nevertheless. So just to summarize a little bit what we're doing on the immuno-oncology, there's a few interesting things that are different from how people have modeled immuno-oncology in microfluidics, because here we control the number of cells, we control the start time and the droplet contents. And this allows us to actually ask different types of questions than in typical organ-on-a-chip experiments. And the analysis method of probabilistic approach is quite original, and it's really linked to this ability to run many experiments in parallel to get probabilities of outcomes. And then by combining the experiments and the analysis, we're really able to learn new biophysics, even though we haven't looked at the molecules themselves. We find the signature of new biophysical processes. So general summary, what we tried to show you today is how to do two-dimensional droplet microfluidics. We went through it very quickly, but this is a robust, easy to fabricate type of microfluidics, and it allows parallel operations and observations in many droplets. Making spheroids inside of this adds a little bit of complexity. You have to manage the surfactants, the cell culture media, the size, and so on. But I think that if you're used to doing cell culture, there is no real barrier anymore to doing this. We tend to publish the details of our experimental protocol, so it should be easy to copy. And maybe the bottleneck right now is in the image analysis and data analysis, because we end up generating lots of data, hundreds of thousands of cells, tens of thousands of spheroids. So we have to develop the image analysis pipeline better, but this is something we're working on actively. And then I like this little nose on the anchors, because it allows you to do a lot of different things on the droplets. You can add things, you can change from liquid to solid, you can change the contents of the droplets, and this really opens a lot of opportunities. And so just to really finish, most of the work that we've shown today has been published since 2017 in these journal articles. It's mostly the work of Raphaël Tomasi and Gabriel Amsela and Sébastien Sarr who developed the initial system. And then more recently, Gustave Rontex, Chain, Adrien Sacerdos have developed the applications. And we do this in collaboration with many different groups at Pasteur, and the main ones are Anna Comano's group and Philippe Rousseau's group. And that's it. Thank you very much for your attention. I see there is one more question, so maybe I will take it. have you considered looking at the embryo to blastocyst problem in the IVF applications? No, we haven't looked at the very early stages of embryogenesis, but we've been talking to some people here in Europe who are working on similar problems, and potentially this is something that we will do in the coming maybe 2 or 3 years, or 4. Yeah, so thank you very much for your attention. I don't know if there are any more questions. Looks like no.
We'll wait probably a couple of minutes for any questions.
We've been a little bit long, but
No, it was some time, and yeah, it's very interesting. Starting from the microfluidic tools to applications in biology.
okay, so
yeah, so it looks like no more questions. So thank you, Charles, once again.
thank you for the invitation. I hope
for accepting our invitation and for a very interesting talk.
Great, thanks for the invitation. Maybe one day we will meet in India.
Yeah, sometime. Yeah.
Okay.
Thanks.
Bye.